Hello, everyone. Thank you very much for joining us at this LabRoots webinar sponsored by Beckman Coulter Life Sciences. My name is James McCracken, and I am a commercial product manager with Beckman Coulter Life Sciences. And I'm very happy to introduce our speaker for today, Luis Allende, Dr. Luis Allende, who's the head of the Cellular Immunology Unit at the University Hospital 12 de Octubre in Madrid, Spain. Dr. Allende is going to talk to us today about some of his research using cell sorting for the study of lymphoproliferative disorders in inborn errors of immunity and hematological malignancies. And so without further ado, I'd like to pass it over to Dr. Allende. Thank you very much for joining us, Dr. Allende. Really looking forward to hearing about your work.
Okay, thank you, James. Hello, everyone, and welcome to this webinar. It's a pleasure to me to show my experience in the study of some lymphoproliferative disorders related to inborn errors of immunity and hematological malignancies. Briefly, inborn errors of immunity or primary immunodeficiencies are heterogeneous genetic diseases altering innate and/or adaptive immunity. Inborn errors of immunity present clinically as increased susceptibility to infections, autoimmunity, autoinflammatory diseases, allergy, bone marrow failure, and/or malignancy. Genetic variants cause diseases by altering the encoded gene product, such as by abolishing or reducing protein expression and function, or modifying the protein to acquire gain of function. Although inborn error of immunity are considered rare diseases, the number of this type of disorders have been growing year by year, and more than 485 genetic defects are related to primary immunodeficiencies and currently described. Inborn error of immunity are categorized into 10 tables, segregating groups of disorders into overlap phenotypes. These tables describe Group 1, combined immunodeficiencies affecting cellular and humoral immunity. This group includes the most severe clinical phenotype in primary immunodeficiencies, known as severe combined immunodeficiency, also commonly known as bubble boy disease. Group 2, combined immunodeficiencies with syndromic features. For example, Wiskott-Aldrich syndrome, or ataxia-telangiectasia, or DiGeorge syndrome. Group 3, predominantly antibody deficiencies. This group includes the largest number of patients as common variable immune deficiency or agamma-globulinias. Group 4, diseases of immune dysregulation characterized by autoimmune cytopenias as autoimmune lymphoproliferative syndrome or hemophagocytic lymphohistiocytosis. Group 5, congenital defects of phagocytes as congenital neutropenias. Group 6, defects in intrinsic and innate immunity as Mendelian susceptibility to mycobacterial disease, or for example, TLR7 deficiency in severe COVID-19 infection. Group 7, autoinflammatory diseases as recurrent fevers. Group 8, complement deficiencies as C1 inhibitor deficiency in hereditary angioedema. Group 9, bone marrow failure as Fanconi anemia and dyskeratosis congenita. And finally, group 10, phenocopies of inborn errors of immunity that they can be divided in associated with somatic mutations or associated with neutralizing autoantibodies. Due to the genetic origin of most of these diseases, symptoms usually appear at the first stage of life, and diagnosis is commonly made during childhood. This early diagnosis allows the establishment of a tailored treatment and appropriate genetic counseling to achieve considerable improvement and even cure these patients. Overall, 60% of patients initially presented with infections only, but other 9% with immune dysregulation only, and 9% with a combination of both infections and immune dysregulation. Two-thirds of patients with primary immunodeficiency presented before the age of 6 years. Nevertheless, some patients with pathological variants are diagnosed in adulthood because of a late onset of symptoms or non-specific multisystemic presentations that finally give rise to a delay in ordering specific molecular studies for the diagnosis of inborn error of immunity. There are different hypotheses to explain the symptomatic delay of these diseases, such as contact with a specific environmental trigger, the low penetrance of some hypomorphic variants, the development of epigenetic modifications of specific genes, or maybe the presence of somatic variants. In this work from our group, we could see in a retrospective study that 25%, a quarter percent of patients were diagnosed in adulthood. As we could see before, inborn errors of immunity are a diverse group of monogenic disorders of the immune system due to germline variants in genes important for the immune response. Over the past decade, there has been increasing recognition that acquired somatic variants present in a subset of cells can also lead to immune disorders or phenocopies of inborn errors of immunity. The discovery of somatic mosaicisms causing primary immunodeficiency has largely arisen from investigation of sporadic cases of primary immunodeficiencies with predominant symptoms of autoinflammation and/or autoimmunity, in which germline disease-causing variants are not detected. Disease-causing somatic mosaicisms has been identified in genes that also cause germline inborn errors of immunity. You can see such as FAS in the slide, and autoinflammatory disorders. There are some challenges in detecting low-level somatic variants, and it is likely that the extent of the somatic mosaicisms causing primary immunodeficiency is largely uncharted. In my department, in the Cellular Immunology unit, we are responsible for the immunological study of patients with suspected secondary or primary immunodeficiencies. For this purpose, flow cytometry is a very important tool in our daily work. Firstly, we obtain percentage and absolute counts of T lymphocytes, including the CD4 and CD8 subpopulations, B lymphocytes, and NK lymphocytes. In some cases, an extended immunophenotype is necessary. For example, due to the clinical phenotype of the patient or due to a pathological TB NK study. An extended T CD4 immunophenotype can be ordered. You can see in the right of the slide, in the right part of the slide. An immunophenotype of CD4 T cells. This gives rise information about naive, central memory T cells by using anti-CCR7 and CD45 array monoclonal antibodies. T regulatory cells using FOXP3 and CD25. Th1, Th2, and Th17 immunophenotype using CXCR5 and CD45 array markers for gating T follicular helper cells and CCR7 and CXCR3 markers for Th1, Th2, and Th17 phenotype. In the left part of the slide, you can see an extended T-CD8 immunophenotype, immunophenotyping of CD8 T cells can give information about naive, central memory, exhausted T cells by using CCR7 and CD45 array markers, activated T cells using markers as HLH-DR in CD8, a senescence phenotype using the marker CD57. Okay, in patients with a suspect of humoral immunodeficiency, an extended B immunophenotype can be ordered. This is the case of a patient with a common variable immunodeficiency, where a switch memory B cell compartment is very low, as you can see in the histogram, compared with a healthy control. This is the immunophenotype of an adenosine deaminase deficiency with a T-, B- phenotype compatible with a severe combined immunodeficiency. This is the immunophenotype of an CD3 delta deficiency with a T-, B+ phenotype. We can see a low CD3 median fluorescent intensity, low CD8 cells, absence of naive T CD4 cells, absence of recent thymic emigrants CD4 cells, and finally, absence of naive T CD8 cells. Patients with a clinical and immunological phenotype compatible with an inborn error of immunity or primary immunodeficiency are selected for a targeted gene panel or whole exome sequencing with the genes included in the classification. The Sanger method remains the gold standard for validation of variants identified by next-generation sequencing methodologies and for familial segregation studies. Finally, functional validation are mandatory in cases of variants of unknown significance, or if the prediction of a new variant is pathological and it has not been reported. For example, immunophenotyping, protein expression, complementation experiments, antigen-specific memory B and T-cell generation, immune cell function, signaling pathway analysis. Anyway, primary immunodeficiencies are a large and heterogeneous group of diseases with a diagnosis rate of approximately 50%. We can find several challenges in the diagnosis of primary immunodeficiencies. A clear phenotype-genotype associations are rare in primary immunodeficiencies due to the heterogeneous and complicated clinical, immunological, and genetic nature of the disease. The heterogeneous presentations of several primary immunodeficiencies in terms of age of onset, severity of disease, and penetrance suggest that other mechanisms, as somatic variants and epigenetics, may play a crucial role in driving the disease. These investigations will require the collaboration with highly skilled experts to enable design of experiments to gain understanding of the role of somatics or epigenetics in PID and whether these modifications represent therapeutic targets of disease. Variants of unknown significance should be confirmed by using personalized functional studies, as we saw in the last slide. And in cases with no germline candidates, whole genome sequencing could be an alternative. I would like to show some applications in our Department of Immunology where cell sorting can make a decisive contribution to diagnosis, validation of molecular variants, or treatment monitoring. Firstly, I will show the diagnosis strategy for identification of somatic mutation in a somatic autoimmune lymphoproliferative syndrome. Secondly, we will see a functional validation of a germline mutation in a deficiency of TET2. And thirdly, the discovery of somatic mutations in new genes in chronic NK-LGL leukemia can help to identify new molecular subtypes and personalize the treatments. According to the European Society for Immunodeficiencies, primary immune regulatory disorders represent approximately 5.3% of primary immunodeficiencies and currently comprise 50 disease-causing genes divided into 4 categories: hemophagocytic lymphohistiocytosis, susceptibility to Epstein-Barr virus, syndromes with autoimmunity, and immune dysregulation with colitis. Autoimmune lymphoproliferative syndrome, ALPS, a well-known syndrome with autoimmunity, is a primary immune regulatory disorder characterized by chronic and benign lymphoproliferation, autoimmune manifestations, and an increased risk of lymphoma due to a defect in lymphocyte apoptosis. Clinically, ALPS patients are characterized by lymphoproliferation, autoimmunity, and malignancy. Expanded CD3 positive, TCR alpha beta positive, CD4 negative, CD8 negative, double negative T cells, these cells are known as DNTs, are a hallmark of all patients who can also present elevated plasma biomarkers, including interleukin-10, soluble Fas ligand, and vitamin B12. Genetic causes comprise the apoptotic Fas ligand pathway components as Fas, Fas ligand, FADD, Cas10, and Cas8. Being germinal and somatic mutations in Fas gene, the most common cause of ALPS. However, approximately 20% of patients with ALPS do not have an identifiable genetic mutation. 70% of all patients present mutations in these genes that you can see in the slide. 15–20% of all patients, it cannot be identified a germline mutation, but we can identify a not inherited condition. It refers to all patients with somatic mutations in the Fas gene. In the rest, in the right part of the slide, in the rest, 15–20% of all patients, we cannot identify a germline of somatic mutation. The molecular study of somatic mutations in the Fas gene is mandatory in patients with elevated DNTs and biomarkers and in whom a germline mutation has not been identified. Clinically, ALPS somatic Fas and ALPS germline Fas patients are indistinguishable. At laboratory level, both conditions share high counts of DNT cells and high levels of ALPS biomarkers as vitamin B12, cellular Fas ligand, and IL-10. Apoptosis in PBMCs of somatic ALPS patients is observed. You can see compared with a healthy control. And the main difference is that the mutation is not found at germline level because the mutation is confined to DNTs in the Fas gene. This is an example of a baby. He was 1 year old when he was diagnosed with high ALPS biomarkers and DNTs, where no germline variants and ALPS-related genes were identified. In this case, a somatic mutation should be ruled out. The patient showed a somatic mutation in Fas gene. In whole blood, the mutation is represented at 4% and only has 10 reads. The candidate variant was validated by Sanger sequencing in a sample of DNA isolated from DNT cells by sorting, as you can see here, and confirming the mutation shown by NGS in this +2 position. Here you can see the double peak in comparison to the healthy donor. In this case, we could demonstrate a functional deficiency of apoptosis in these isolated DNT cells in the somatic ALPS patients compared with a healthy donor. Therefore, we could diagnose a somatic ALPS patient in a child with only 1 year old. A key message is that patients with the somatic form of ALPS can be underdiagnosed. It is important the collaboration for sorting of DNT cells in the laboratory. In this other branch of the tree, we have to consider an ALPS-like phenotype, where we have ruled out large deletion and candidate variants in the promoter of Fas gene. Now, we have in this point of the talk. Advances in NGS technologies have enabled an expanding list of monogenic defects in primary immune regulatory disorders. Numerous genetic defects outside of the Fas ligand pathway, mimicking an ALS phenotype, have been identified as ALS-like syndromes. Clinically, autoimmunity and lymphoproliferation is a common finding in these ALS-like phenotypes. To date, more than 15 genes have been related with ALS-like syndrome in the literature. Phenocopies of PID as NRAS and KRAS deficiencies, susceptibility to EBV virus as MACT1 and PRKCD, regulatory T-cell defects as CTLA4, LRBA, STAT3 gain of function, and this group is the biggest one. Combined immunodeficiencies as ITK and STK4, other defects in intrinsic and innate immunity and predisposition to infection, and finally, autoimmunity and autoinflammation defects as ADA2 and TET2 in the right part of the slide. In this part of the talk, I would like to mention TET2. TET2 is an enzyme belonging to the TET family that acts as a key regulator for gene expression through DNA demethylation, oxidizing the methyl group of 5-methyl cytosine, known as 5-MC, to 5-hydroxymethyl cytosine, known as 5-HMC. TET2 is mainly expressed in hematopoietic cells, where plays important roles, including self-renewal, lineage commitment, monocyte differentiation. Recent studies have evidenced the importance of TET2 also during immune cell differentiation, activation, and immune homeostasis. The first human molecular variants described in TET2 with clinical implication were reported in 2009. These variants are somatic variants and show implications in hematological malignancies. After that, the first monoallelic germline mutations, also implicated in hematological malignancies, were reported in 2019. And finally, biallelic germline mutations were reported in 2020. This form of the disease is autosomic recessive, and clinically, patients present an ALS-like phenotype and lymphoma. Recently, our group have reported the fourth patients with autosomic recessive inheritance form of TET2 deficiency. The patient suffered two lymphomas, the first one in 2010 and the second one in 2020. An ALS-like phenotype characterized by lymphadenopathies, hepatosplenomegaly, and cytopenias. The clinical and analytical phenotype presenting with lymphoproliferation, cytopenias, and immune dysregulation led us to suspicion of an ALS syndrome. A matched and related donor hematopoietic stem cell transplant was performed in March 2021, but unfortunately, the patient died early due to transplant-related toxicity. The immunological description of the patient is shown in this slide. Panel A shows two different mutations in TET2 gene, in the index case P1. The mutation configuration in trans confirm a biallelic form of the disease. The familial segregation study revealed the presence of a mutation in the younger healthy brother, HC, while no mutations were identified in her mother and older brother. Although her father had previously died at the age of 55 years and he could not be analyzed, we assume that the shared mutation between P1 and HC in dark blue was of paternal origin, while the other in light blue was a de novo mutation. Panel B shows an immunophenotype with high numbers of DNT cells and some high ALS biomarkers as vitamin B12 and interleukin-10 in the patient. The healthy P1's brother, or HC, carried a TET2 mutation and presented some markers of immune dysregulation as high levels of IL-10. In panel C, in the left part of the Western blot, we show the lack of protein expression, confirming the biallelic form of TET2 deficiency in this patient. We wanted to demonstrate the importance of these mutations in the functionality of TET2. Global DNA methylation was evaluated by relative quantification of genomic 5-MC percentage if DNA extracted from whole blood of P1 and her healthy carrier, HC. In DNA samples from whole blood, we measured the levels of 5-MC as a functionality marker of TET2 activity. In other words, the level of methylation. As we can see, P1 had high levels of 5-MC, and we could conclude that the functionality of TET2 was impaired. By the contrary, the level of 5-MC in her healthy carrier was normal compared with several healthy donors. Mutations in TET2 are considered a pregmalignant event, and the co-occurrence with other cumulative mutations can trigger the lymphomagenesis. This is the risk that we want to test in the healthy carrier. Furthermore, lower activity of transcription factors involved in development, activation, cytotoxicity, and exhaustion of CD8 T cells and NK cells has been reported in TET2 mutations carriers, maybe implying dysregulation of cytotoxic lymphocyte maturation and function. Here you can see on the right the high level of TENRA CD8 T cells and the high expression of the senescence marker CD57 in the carrier of TET2 mutation. For this reason, using this gating strategy, we sort TENRA CD8, naive CD8, and NK cells. We select the enriched mode for sorting because the recovery was the most important aspect of the sort. Firstly, we select lymphocyte using forward and side scatter. We select singlets. We select CD3 positive cells for CD8 sorting and CD3 negative for NK sorting. With this condition, you can see the recovery of cells of the three populations, in this case, Tembra CD8 cells. In this other slide, you can see naive CD8 T cells and finally NK cells. To evaluate if DNA demethylation of the TEC2 function was altered in these cells, in a healthy carrier, the percentage of 5-MC was quantified in isolated cells populations as PBMCs and sorted NK, naive, and Tembra CD8 T cells that we saw before. P1, as we knew, shows significantly increased percentage of global 5-MC in whole blood, confirming the functionality consequence of TEC2 deficiency, while no alteration were found in her heterozygous carrier HC. However, when methylation levels when compared between the healthy carrier HC in dark blue and healthy donors in light blue, in sorted cell populations, healthy carriers show higher levels of 5-MC in all cell populations compared with the healthy donors, with a statistically significant difference in Tembra CD8 T cells. So the healthy carrier presents a TEC2 deficiency only in some lymphocyte subpopulations. So with these results, we decided that the healthy carrier should be monitored in the immunology department. Okay, in the last part of the talk, I will show the use of cell sorting in the case of lymphoproliferative disorders as a large granular lymphocyte LGL leukemia. LGL leukemia is an indolent lymphoproliferative disorder characterized by a clonal expansion of cytotoxic T or NK lymphocytes. Pathogenesis is still incompletely understood. Recent molecular discoveries involving the STAT3 activation pathway have improved comprehension of the disease. Treatment is based on immunosuppressive therapies, which produce an unsatisfactory long-term response, making targeted therapies the next step to a curative approach. LGL leukemia comprises approximately 2 to 5% of all chronic lymphoproliferative diseases diagnosed in the US and Europe, with a slightly high prevalence in the Asian population. The disease is most commonly diagnosed in elderly patients with a median age at presentation of 50 to 60 years. It equally affects men and women, but the diagnosis in women is often made at a younger age. The median overall survival is 9 to 10 years, and only 10% of patients are younger than 40 years, and pediatric cases are sporadic. The two subtypes of chronic LGL proliferations are T-LGL that represents 90% of cases, and NK-LGL that represents 10% of cases. Clinically, LGL leukemia harbors an indolent presentation, cytopenia, and autoimmune-associated conditions. STAT3 constitutive activation is the hallmark of LGL leukemia, with STAT3 mutation found in 40 to 70 of patients. Diagnosis is based on expanded clonal LGL cells harboring a constitutive mature posthemic phenotype. Treatment is required in 60% of patients at diagnosis and is based on immunosuppressive therapies as methotrexate, cyclophosphamide, and cyclosporine A. New specific targeted therapies based on JAK-STAT inhibitors or cytokine inhibitor are being tested with promising results. The pathogenesis of LGL leukemia is firstly characterized by proliferations related to chronic antigen stimulation leading to a polyclonal expansion. Clonal proliferation is sustained mainly by interleukin-15 and pro-inflammatory cytokines promoting natural killer cell or leukemic LGL survival. The hallmark of LGL proliferation is the constitutive activation of STAT3. This activation is explained by common somatic gain of function STAT3 mutations in about 40% of patients with LGL leukemia. Moreover, LGL leukemia cells are resistant to Fas-mediated apoptosis. Soluble Fas acts as a decoy receptor able to inhibit Fas-dependent apoptosis. Other mutations in other genes are also implicated in the pathophysiology of LGL. This is the case of a 61-year-old man followed up in Hematology Science 2018 for mild to moderate chronic thrombopenia and neutropenia without hemorrhagic manifestations at any time. He was diagnosed of Hashimoto thyroiditis before the diagnosis of LGL. A splenectomy was performed due to massive splenomegaly and persistent lymphocytosis. The diagnosis was LGL leukemia with a very high number of total lymphocytes due to a very high expansion of CD8 T cells and NK cells. Here you can see the CD8 compartment represents a mature senescent exhaustion phenotype with high percentage of effector memory cells and TERA on the left, very activated with very high expression of HLA-DR and exhausted and senescent phenotype with high percentages of PD-1 and CD57 on the right. The patient has two variants in a DNA sample from peripheral blood. A pathogenic variant in STAT3 gene with a variant allele frequency of 30%, causing a missense threonine to methionine in position 716. This mutation was previously described as pathogenic in germline STAT3 gain of function patients. A second missense variant in TET2 gene was obtained with an uncertain significance and variant allele frequency of 3%. The study of molecular variants in these genes shown, such as STAT3 and TET2, and other in LGL leukemia, is interesting for a better understanding of the pathophysiology of these diseases, and also could correlate with the personalization of immunosuppression. The follow-up of variant allele frequency in the different lymphocyte population studies, such as CD8, CD4, TCR gamma delta, or NK lymphocytes, and the correlations with clinical markers as neutrophils, platelets, hemoglobin, can help to optimize the treatment in these patients. The study of molecular variants in different lymphocyte populations can help in the future in the clinical follow-up of these patients and in the monitoring of new immunosuppressive therapies specific for a lymphocyte population. For example, CD8 in case of T-LGL or NK in case of NK-LGL. This is the gating strategy for sorting CD4, CD8, and TCR gamma delta T cells, and CD3 minus, CD16 positive NK cells. Here you can see the purity of CD4 and CD8 sorted T cells. And here the CD3 negative, CD16 positive NK cells, and below the CD3 positive, TCR gamma delta positive T cells. With sorted cells, we will test both variants in the patient by NGS, the variant in STAT3 and in TET2 genes. If the efficacy of the treatment is correct, we will see a correlation between the variant allele frequency and absolute lymphocyte counts, CD8 and NK cells, and neutrophils, platelets, and hemoglobin. As conclusion, cell sorting can be important in an immunology department for diagnosis. Isolation of double negative T cells for identification fast mutations to diagnosis a somatic autoimmune lymphoproliferative syndrome. Treatment monitoring, isolation of CD8 memory T cells and other subpopulations can help to demonstrate an altered epigenetic function in individuals with germline or somatic variants in TET2 genes that can trigger lymphomagenesis and malignancy. Furthermore, isolation of CD8 and NK lymphocyte population for identification somatic variants in genes as STAT3 and TET2 to understand the biology and genetic abnormalities of LGL leukemia. Changes in variant allele frequencies can assess the effectiveness of treatment. And many other applications related to diagnosis and research can be used in an immunology department and other specialties. And finally, I would like to thank to my collaborators, especially Celia, Marta, and Maria Jose in my hospital 12 de Octubre, and other persons from other centers, and public and private sponsors. Thank you very much for your attention.
Okay, great. Thank you very much for that, Dr. Allende. Very much appreciate your time to present this to us. I did have a few questions that had come in. I'd like to ask you if that's all right.
Okay, I'm ready.
Okay, I noticed you got some pretty pure populations at the end there in your sorts. I wonder, do you have a feeling for how many cells you need and what purity you need to support your further molecular assays?
Okay, in our experience, my team has sorted more than 200 human samples. All samples were from human whole blood, okay? The first step was the peripheral blood mononuclear cells isolation by FICOL, okay? We usually begin the cell sorting with a minimum of 5 millions of PBMCs, where the main populations are CD4, CD8, CD19, and NK cells. We usually recover between 40 to 50% of cells. I think it's recommended to increase the number of initial cells when your population is less represented. No, we sort using the enrichment mode. No, I think I told, but I remember we use the enrichment mode because with other modes the sorting is more restrictive and the recovery is lower. And with this enrichment mode, we get an average purity of 92, 95% with a standard deviation more or less of 6%. No, and usually sorted samples will be used for DNA isolation. No, I think it's important to say that. No. And with this DNA, we use in molecular studies by next generation sequencing. And in some cases, we use sorted cells for some functional studies like apoptosis test.
Okay, thank you. I saw a question come in. You have a few different pieces of hardware in your lab as far as flow cytometers. Do you find you can use the same sample from your other flow-based tests for sorting, or do you prepare a sample only for sorting and only for analysis?
Normally, we use the sorting only for sorting of cells, but we have an exception. No, for example, sometimes we use the sample at the same time for diagnosis and research at the same time. This example is the case of all patients where the same preparations can be used for the counting of DNT cells and for isolation of these cells.
Okay. And I think this question is less a technical question and maybe where you think things might be headed next. When you find a variant that's of unknown significance, what do you do next?
Okay, I think this question is related to Himbo errors of immunity. No, functional validation are mandatory, or if the prediction of a new variant is pathological and it has not been reported. No, in the first step, we usually use flow cytometry to test lymphocyte populations, or even the protein, if we have an available monoclonal antibody. No? The second step usually is to show if gene expression is affected by using RNA gene expression or Western blot techniques, no, as we saw in the presentation. And my group is specialized in diseases of immune dysregulation, and for us it's important to test lymphocyte apoptosis, no, as you saw in the presentation, using annexin 5 monoclonal antibodies and propidium iodide. And it's important for us to demonstrate if the patient has an apoptosis deficiency, that we can correlate, for example, with a variant of an unknown significance. No? Furthermore, if gene expression is affected, then normally we are in contact with other groups of research, no? And this is important to collaborate with highly skilled experts to enable design experiments to gain understanding of the role of this variant of unknown significance, and if it's involved in the pathophysiology of the disorder.
Okay. All right. Well, thank you very much again, Dr. Allende. I think with that, we're going to end this webinar today. I'd like to thank all of our attendants once again, and thank you to LabRoots for broadcasting this, and wish you all a good day, and see you next time.
Okay, thank you very much for your attention. Thank you. Bye.
Thank you. Okay. Great. Thank you very much for that, Dr. Allende. Very much appreciate your time to present this to us. I did have a few questions that had come in. I'd like to ask you if that's all right.
Okay, I'm ready.
Okay. I noticed you got some pretty pure populations at the end there in your sorts. I wonder, do you have a feeling for how many cells you need and what purity you need to support your further molecular assays?
Okay, in our experience, my team has sorted more than 200 human samples. All samples were from human whole blood, okay? The first step was the peripheral blood mononuclear cells isolation by FICOL, okay? We usually begin the cell sorting with a minimum of 5 millions of PBMCs, where the main populations are CD4, CD8, CD19, and NK cells. We usually recover between 40 to 50% of cells. I think it's recommended to increase the number of initial cells when your population is less represented. No, we sort using the enrichment mode. No, I think I told, but I remember we use the enrichment mode because with other modes the sorting is more restrictive and the recovery is lower. And with this enrichment mode, we get an average purity of 92, 95% with a standard deviation more or less of 6%. No, and usually sorted samples will be used for DNA isolation. No, I think it's important to say that. No. And with this DNA, we use in molecular studies by next generation sequencing. And in some cases, we use sorted cells for some functional studies like apoptosis test.
Okay, thank you. I saw a question come in. You have a few different pieces of hardware in your lab as far as flow cytometers. Do you find you can use the same sample from your other flow-based tests for sorting, or do you prepare a sample only for sorting and only for analysis?
Normally, we use the sorting only for sorting of cells, but we have an exception. No, for example, sometimes we use the sample at the same time for diagnosis and research at the same time. This example is the case of all patients where the same preparations can be used for the counting of DNT cells and for isolation of these cells.
Okay. And I think this question is less a technical question and maybe where you think things might be headed next. When you find a variant that's of unknown significance, what do you do next?
Okay. I think this question is related to hindborn errors of immunity, no? Functional validation are mandatory, or if the prediction of a new variant is pathological and it has not been reported, no? In the first step, we usually use flow cytometry to test lymphocyte populations, or even the protein, if we have an available monoclonal antibody. No? The second step usually is to show if gene expression is affected by using RNA gene expression or Western blot techniques, no? as we saw in the presentation. My group is specialized in diseases of immune dysregulation, and for us it's important to test lymphocyte apoptosis, no? As you saw in the presentation, using annexin 5 monoclonal antibodies and propidium iodide. And it's important for us to demonstrate if the patient has an apoptosis deficiency, that we can correlate, for example, with a variant of an unknown significance. No? Furthermore, if gene expression is affected, then normally we are in contact with other groups of research, no? And this is important to collaborate with highly skilled experts to enable design experiments to gain understanding of the role of this variant of unknown significance, and if it's involved in the pathophysiology of the disorder.
Okay. All right. Well, thank you very much again, Dr. Allende. I think with that, we're going to end this webinar today. I'd like to thank all of our attendants once again, and thank you to LabRoots for broadcasting this, and wish you all a good day, and see you next time.
Okay, thank you very much for your attention. Thank you. Bye.
Thank you.